Hello friends, welcome in the YouTube video class. Myself Professor Mundhe Sandeep, Head and Assistant Professor of Microbiology, Sunjini Mahavidyalaya Sapule, Taluka Sakur District, Latur. We are continuously organizing microbiological classes for students and learners. And this is the class of class for BSc second year, where we are going to study the chemical agents are used to control the air microorganisms. Let us see how these different chemical agents they are used to control the microorganisms. It is very difficult to use liquid as well as a solid form, powdery form of the chemicals to control the air microorganisms in the walls, in the roofs, inside the machineries, or it is very difficult to use to control the air microorganisms. That's why a gaseous form of the as well as vaporized form of the chemicals they are mostly used to control the air microorganisms, and they are important chemical agents to control the air microorganisms, and especially formaldehyde, then beta propionolactone as well as ethylene oxides. They are generally used to control the air microorganisms because they are used inside the chamber, inside the room, they are in the hospital, in the operation theater, in the research centers, or any other places, enclosed rooms, where these gases and vaporized chemical agents they are used to control or to kill the microorganisms which are present within that. So these are very important chemical agents. The first is the aldehyde group, that is the formaldehydes. It is an antimicrobial agent. Why antimicrobials? They are used to kill the microorganisms. They generally inactivate proteins. What is the mode of action of formaldehydes? It inactivate the proteins. Inactivate the proteins means whatever the proteinic nature part of the microbial cells, they are inactivated due to the action of aldehydes. Then it generally what is next mode of actions of the aldehyde group, that is the covalent cross-link formation. Whatever the covalent bond present between the cell, between the proteins, between the any parts, that they generally affect. They are effective chemical agent. They are carcinogenic, and they are useful to oxidize molecules inside the cells. Oxidize means whatever the molecules, whatever the reactions inside the bacterial cell, microbial cells, if they are undergoing oxidations or due to the effect of formaldehyde, if there is the oxidized form, it means that what happens, there is a side effect in the cells. So gaseous sterilizing agent, the chemical reactive gaseous ethylene oxide formaldehyde possesses broad spectrum biocidal activity. Broad spectrum, it means that they are mostly effective against different types of microorganism. And biocidal, it means it kills. It generally, they are carcinogenic type, that's why they generally kills the microorganisms. And they have found application in the sterilization of reusable surgical instruments. Generally in the operation theaters, a number of surgical instruments or surgical equipments are present. These equipments, they are generally sterilized by using the gaseous sterilizing gases, because it is very important to avoid the entry of the air microorganism during the operations of any peoples, because during operation times what happens, whatever the part of the body parts they are damaged due to the damage, or they are cutted, so it is open inside the operation theater, and it is very important to avoid the air microorganism entry inside the body. That's why it is very important to fumigate the room. Fumigation, it is one of the vaporization process where the vapors of that gaseous chemicals are spreaded inside that, and due to which the microorganisms are killed. Generally, in laboratory level, we can also use the fumigation process where KMnO4 and K2Cr2O7 potassium dichromate. These are the two chemical agents they are used, due to the mixing of these two, what happens? The fumigation spreaded within second. The fumes of such a mixture are spreaded within the rooms, within the laboratory, and laboratory they becomes free from air microorganisms. They are free from means they are sterilized. And certain medical diagnostic and electrical equipments and the surface sterilizations of powder, they are sterilized by due to the gaseous sterilizing agent. Sterilization process using ethylene oxide sterilization are far more commonly used on an international basis than those employing formaldehyde, means they are more effective than the formaldehydes. Let us see one by one. First, the ethylene oxide. Recognized sterilization method. This ethylene oxide, it is a gaseous sterilizer. They are generally recognized to use in the sterilization methods in BP and BPC, that is the British Pharmaceutical Codex. This gas is highly explosive in a mixture of less than 3.6% volume by volume in air. It means that we cannot use this ethylene oxide in a concentrated form, but we can use this ethylene oxide which is in diluted form. It means it is a 3.6% volume by volumes, and it is an effective concentration, effective dilutions against the microorganism. In order to reduce the explosions hazards, it is usually supplied for sterilizing purposes as a 10% mixed with CO2. It means that they are generally used in combination with carbon dioxide powder, that is carbon dioxide in vaporized, and due to the combinations of 10% mixed with the carbon dioxide, what happens? There is no side effect of ethylene oxide to humans, and they are effective against only microorganisms. Or 8.6% mixtures with HFC, that we can say hydrofluorocarbon, which has replaced a fluorinized hydrocarbon. It means that the ethylene oxides, they are used in combinations with other compounds. One is the carbon dioxide, or that is the hydrofluorocarbon, which is 8.6%. Alternatively, pure ethylene oxide gas can be used below atmospheric pressure in sterilizer chamber, from which all air has been removed. It means that it is very dangerous, because we can use in pure form ethylene oxide, but if only when the atmospheric pressure is below atmospheric pressure in sterilizer chamber, from which all air has been removed. It means that it is very effective. After that, the next gaseous sterilizing agent or other chemical agent which are used to kill the air microbes, that formaldehyde gas. It is very simple gaseous sterilizing agents. They are generally used in different places. Formaldehyde gas for use in sterilization is produced by heating formalin to a temperature of 70 to 75 degree with steam, leading to the process known as LTSF. It means that whatever the formaldehydes which are vaporized from, created only when at the temperature 70 to 75 degree temperature at the steam, leading to the process known as LTSF. Formaldehyde has a similar toxicity to ethylene oxide. It means that both formaldehyde as well as the ethylene oxide has a similar toxicity against the microorganism. Similar toxicity means they generally kills the microorganism at a specific concentrations. So due to which what happens, we can kill or we can destroy these microorganisms, although absorption to materials appear to be a low, or whatever the effectiveness of these chemical agents. So due to which what happens, whatever the absorptions to materials appears to be a low. That is very important. And whatever the fumigation process, these gases sterilization process, they generally carried out within the room, within the hall, within the laboratory, within the research laboratory, or within the operation theater, or in any industrial level. So due to which these are the area are they are the very sensitive areas, because we can operating a highly important operation, as well as we can carry out a number of practical and research activities, and it must be avoid the entry of the microorganism within the rooms. Formaldehyde is the simplest compound to aldehyde series. The characteristic feature of the compound is that at room temperature it polymerizes and exists as in solid substance, where at in cleavated temperature it depolymerizes to gas. It means there are gases forms appear in the different stage. Apart from that, what happens, we can also use the other chemical agent, that is the beta propiolactones. This is also one of the important gases sterilizing agents we can use in the to sterilize the air micro, to kill the air microbes, that is beta propiolactone. It is also one of the good gases sterilizing agent with wide spectrum of antimicrobial activity, and it is a sporocidal, fungicidal and virucidal. It means that they are very effective against, they are, because due to the use of formaldehyde and ethylene oxide we can kill the microorganism, but we cannot destroy the spores of a specific, because spores, they are the chemical resistant, they are the heat resistant, they are the UV resistant body, they cannot easily destroy due to these. But there are exceptional chemical agents, that is the beta propiolactones. They are generally sporocidal, fungicidal and virucidal. It means that whatever the spores of bacteria, whatever the viruses and fungi, they are destroyed due to the use of beta propiolactone. This compound is colorless liquid at room temperature with high boiling point 155 degrees Celsius temperature, and beta propiolactone is highly effective against microorganisms. Only 2 to 5 milligram per liter of beta propiolactone is required to kill all types of microorganism, and it acts on microorganism by inactivation of protein and other biomolecules. It means that the mode of actions, each and every chemical agent has its role. Its role means each and every chemical agent has its mode of action against microorganism. It means that the specific place of bacteria, specific place of microorganism where these chemical agents are effective. For example, some chemical agents, they destroy the proteins of the microorganism. Some chemical agents, they destroy the nuclear material of the DNA. Some chemical agents, they destroy the cell wall of bacteria. Means whatever the parts of the microorganism, they are destroyed due to the effect of these chemical agents. So this is all about the different types of chemical agents, especially the gaseous form of the chemical agent, vaporized chemical agents, or that is the fumigation process. They are generally used to kill the air microorganisms. So this is all about the different types of chemical agents. They are used to kill the air microorganism. With the end of this part, so the unit, the air microbiology, is completed. So friend and my dear students, in the next lecture we will start the another unit, that is the water microbiology. So with the completion of this part, the topic is over, and I request to all of you, if you are watching this video, if you are attending the daily class, you must read, you must concentrate on this video, and you can carry the you can read other information regarding this topic, and in the at home you have to separately you have to point out what the notes of these node and on these topics, and we'll see your notes after a period of times. Till then, bye.